the first day, I think, that we experienced full single-ton heat. Not looking forward to triple digits, 103 degrees this afternoon. Yes, to the rest of your summer. Yeah, I'm like sweating right now. Is it hot or is it just me? I'm running around.
Did you walk up the stairs?
I did. Jesus, am I not unfit? Bad. So how did yesterday's biochem lecture go for nucleic acids? Amazing. Go for anything a little bit? Okay. Well, we won't talk about much until block 2 with that, but hopefully you have a decent foundation. And no Michaelis-Menten equations required for this course, so whatever you experience this afternoon, we're not going to use that. So the deck is up, like 1,000 apologies for how long it took for me to get this up. I will say though, the original deck was 129 slides, so this is like a 35% reduction. Yeah, much shorter. I think it'll take about 3 days to get through it. We'll go slowly because these slides are pretty dense. There's a lot of material on there, but just slowly, and it's very repetitive and iterative. So we cover a topic that's not clear the first time, circle back, cover it from a different angle, and we should get pretty good comprehensive coverage by the end of this deck. And then of course we still have the review, just whenever next week, but this scheduling is really, really good this semester for block 1. We'll have plenty of time to hit that review and get you a couple days to just sit with the material and really become solid with it. Okay, so I'm not worried about it too much other than the fact it's top immunology. It's a really complicated subject. If I didn't tell you this before, my older brother, he got his PhD in immunology. He was doing research on mycobacterium tuberculosis, and specifically like how that activates macrophages. And when he was getting his PhD, I was an undergrad at the time. He used to call me up and just bore the crap out of me with all this research and like how many mice he was sacking, all this stuff. I was like, oh my god, I never want to go into immunology at all. And then after he got his PhD, he like 6 months later he went to PA school, and now he's a slave PA. How crazy is that? Wow. The irony.
What do your parents do?
They're truck drivers.
Heck yeah.
Yeah.
Hell yeah.
That's how they met. They met in truck driving school in South Prairie, Wisconsin. Super romantic. No, we're biological children. Yeah. But if you PubMed the last name Holzberg, you'll see like up until around 2010, all those publications are my brother's, and they're all in like tuberculosis. And then after that, it becomes like my publication record. So yeah, I mean, I was kind of hoping he would stay in research so we could, publish together someday. But now he's a PA, like everyone else in my life is a 65 Delta. Boo. What's that? Okay, thank you.
Who's the favorite?
My brother, for sure, 100%, yeah. I mean, because so he's a civilian PI, I forgot to say that, but he got really PA school in Iowa, University of Iowa. And then he moved back to Madison, and he had a kid, and he has a family, so it's really nice for my parents because everyone is in Madison. And I, never call, I never visit. I'm always busy. I'm going home, though, not next weekend, but the weekend after that, the Memorial Day weekend. So, it's my first time back in Madison in, I don't know, a year or two, but I don't get home very often. I love Madison. It's such a happy place. If you haven't been, and if you consider one of those, the entire state to be a flyover state, blah, 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 you should go check it out. It's a lovely place. Yes.
Sorry.
No.
How was that 70 show received in Madison?
I have no data to answer that question. I don't know. Is it like Wisconsin? Does it have Wisconsin ties?
It's in Madison.
No way. Oh, thank you. Yeah, my pop culture background is scant. It's lacking. I did not know those things. But yeah. So before we dive into this craziness, do you guys want to handle the review questions from yesterday? Yeah. All right. Let's do that. So many animations I forgot about. Okay, So this is question one, what is endomycorrhizae? You guys remember the term for bacteria that just saprophytic bacteria, yep, saprophytes. Good job. Most of these are pretty straightforward first order questions. So it's a little bit more involved. When we're trying to grow up bacteria, what are some things we should consider in general? Temperature,
Medium. Medium, salt,
types of media, and the nutrients in the media. The salt is important. They don't. A lot of them aren't very salt tolerant. Moisture, humidity, exactly. Yep. What about yes, sir? pH, yeah, super important. Yeah, they don't tolerate super acidic or super basic conditions. Most of them. And time, right? Some of them take a long time to grow. Very good. A couple just basic questions about atmospheric conditions and what terms we use for them. What about the bacteria that like atmospheric concentrations of oxygen? What are those called? Aerobes, aerobic bacteria. What about ones that prefer reduced oxygen? Microaerophilic. Yeah, they can't handle as much. Those that absolutely cannot grow in the presence of oxygen. Anaerobics, yeah. If they cannot grow, then they would be strict or obligate anaerobes. And then what about the ones that take it or leave it, can do with or without oxygen? Facultative, exactly. Facultative anaerobes. There was another term for bacteria that like increased carbon dioxide levels. Hapnophiles, very good. And then we didn't go into this in a lot of detail. We will, don't worry. But how about maybe two characteristics of atypical bacteria that we talked about that are useful in distinguishing them from typical bacteria? They're smaller, and their cell walls different. Take that. Excellent. All right. Fungi. Moving on. If you were asked, if you were given this description, what would you say? Unicellular fungi with a size range. Yeasts. Exactly. Yep. Molds. If they're multicellular, they're molds, multicellular fungi. The size range of viruses that we talked about.
20 to 300.
Yes. What was it? Units. Was it nanometers? Yeah. And how? What fraction of a meter is a nanometer? 1,000. It's a billion. Yeah, so micrometer is super small. Yeah, so we've got a centimeter, hundredth, we've got a micrometer, millionth of thousandth is blanking. But yeah, a millionth is a micrometer, and then a billionth is a nanometer, and a trillionth is a picometer. So, and picometer would be in the size range when we're talking about like atoms, or very, very small molecules. Okay, so the size range is 20 to 300 nanometers for viruses for the most part. True or false? Viral genomes have one circular haploid double-stranded DNA chromosome. That's false. It's wrong on so many levels. That's basically the definition for bacterial genome. They usually do have one, but some of them have multiple segments of their genome. Some of them are circular, some of them are linear. We didn't talk about that, but they're not all circular. They're usually haploid in that they have one copy of those genes. HIV packs two exact copies, but that's an unusual one. Double-stranded, we talked about, you can have either double-stranded or single-stranded genomes, and you can also have RNA genomes as well. And we don't usually call them chromosomes. Chromosomes usually refer to cellular genomes. With viruses, it's just their nucleic acid genome. So lots wrong with that statement. True or false, RNA viruses replicate in the cytoplasm and DNA viruses replicate in the nucleus for the most part. True statement. Yeah. True or false, viruses must produce their own ribosomes for protein synthesis. That's false. Yeah, very good. Yeah, we just use ours. When we talk about the viral replication steps when those viruses are ready to leave the host cell, we talked about some different ways that they do that, different ways between envelope viruses and non-envelope or capsid viruses. How are they different?
One's first through the cell, and the other one just gets enveloped in the capsid.
Yeah, I like it. Yeah, good. Okay, so if you're an enveloped virus, something like a paravoxovirus, Ebola virus, measles, something like that, an envelope virus is going to what? Is it going to bud or is it going to kind of violently explode? It's going to bud gently, like a normal exocytosis process from the host cell. It's like a membrane budding. So well tolerated relative to capsid viruses, which leave by what? It kind of just lyses cell. Yeah, it's game over with the cell when you get enough of those where they burst the cell. We talked about the eclipse phase of viral infection. What's going on during the eclipse phase? Sir? Yeah, all the biosymbiotic events. So it takes time for them to replicate to sufficient numbers where you have a condition what we call viremia, enough viruses to actually do stuff. So the eclipse phase is when it is busy making copies of itself in the host cell. We haven't, I don't think we've defined this word before, etiologic agent or etiology. What does that mean? Yeah, exactly. If I ask you what the etiology is of COVID, you could say SARS-CoV-2. Yeah. So the etiologic agent of prion diseases, what's the nature of that agent? It is a misfolded protein, exactly. PRP-C, C for cellular, prion protein cellular. And how does it amplify? Does it replicate? Does it divide? Like, how does that propagate? Yes, sir.
Doesn't it come in contact with other proteins and start to misfold those proteins as well?
It does. That process is called conversion. But when it contacts other proteins, is it just any protein, or is it specific?
That's a good question.
There's other prion proteins, other normal cellular prion proteins, and then they become misfolded, and then they become that SC prion protein scrapey. But good, great. So that is conversion. What part of the body do prionopathies or diseases caused by prions affect? The brain. And then just another term that we were kind of throwing around yesterday a little bit, that we'll continue to use a lot in this course, is zoonotic diseases, zoonosis. What does that mean? Exactly, a disease that can be passed from animals to humans. The 4 types of motility are commonly applied to protozoan parasites.
Flagella and
Ciliary.
Yeah, you do this by interpretive dance, right? Flagella.
was that globular, whatever?
Cilia.
What's the other one?
What is the other one? What's the one that's kind of, do your
I don't know, it's
interpretive dance.
globulus something.
So we have flagellates, we have cilia, ciliates, and then we have another one, amoeba. They move by amoeboid movements, their cytoplasm, external projections. And then there was a 4th,
They're like non.
yeah, kind of the non-modal ones, it's like a gliding motility, but it's not much body flexion basically. Well, what are they called? Apicomplexans, exactly. So flagellates, ciliates, amoeba, and apicomplexans. We talked about 2 different forms that protozoans can take. You guys won't remember what the names of those 2 different forms are? Cysts and trophozoites, or tropes for short. What's the difference between those 2? Yeah, trophozoites are metabolically active. They're the ones that can replicate. They're the ones that can eat. And cysts are the ones that are basically like hardened up for survival and transmission from person to person, not metabolically active, not much going on. Okay, and then this is a kind of a hard one, but when we were talking about worms, which are also known as, what word that starts with an H?
Elements.
elements, yeah.
Cestoids are
We talked about three different groups.
tapeworms.
Cestodes are tapeworms, trematodes are flukes, and roundworms are roundworms. Good, okay. Okay, cool. And any questions for anyone yesterday?
I have a question.
Yeah.
So I did some research last night, and a part of it came to like when a virus comes in contact with the cell, and it transfers this like genetic information into the cell.
Mm-hmm.
I can't remember the name of the video, but it was talking about how it stays in that cell until the cell goes through mitosis, and then it replicates that virus's DNA, and then at some point it just decides to kill the cells, all of the cells after all of the mitosis. I was wondering what that was called.
That sounds like a latent DNA virus. Do you remember the name of the virus that was talked about in that video?
I can try and find the video again.
Yeah,
Send it to you.
that would be interesting. So there are a couple different ways to classify different types of viruses, but lytic viruses are the ones that we talked about that really don't care what's going on in the cell. Like the RNA viruses will hit the cytoplasm regardless of what stage in the cell cycle that cell's in, and they'll just replicate. DNA viruses that are lytic just do their business too and leave the cell. But if a virus is capable of establishing latency, like the herpes viruses, like herpes simplex, cytomegalovirus, Epstein-Barr virus, all those viruses, or HIV, then there's, once that DNA genome gets into the nucleus, that's after it's encoded and been translocated into the nucleus, it can actually integrate into the host nucleus, that entire DNA genome, and it can stay what we call quiescent, or stay integrated, stable, quiet, for long periods of time. So that sounds probably like.
That's how it was explained.
Yeah.
Whenever it exits the cell, is it in that lysis form or is it in that like exocytotic form?
Yeah, so once it gets a signal, a trigger for reactivation, it becomes transcriptionally active and it starts producing, it basically goes back on track for a lytic style of replication cycle. So it starts producing progeny virions that will start doing damage, start drawing attention from the immune system, all that stuff that doesn't happen when it's latent in the genome.
Okay, so all those cells just die at once.
No, they don't necessarily. So like for example, with both HIV and herpes, when they get reactivated, they might start producing considerable amounts of progeny virions. But these are envelope viruses, so they're going to be budding kind of gently and gradually out of those host cells. And particularly with HIV, those host cells can sustain that level of budding for long periods of time. It's a slow burn infection. You know, you produce a lot of CD4 T cells, which is the primary target of HIV. So there's a lot to infect, but even when it's going and transcriptionally active, you're not going to be killing huge amounts of CD4 cells. You'll kill a lot, but not like rapidly depleting the entire body supply. So, great question, and we will definitely cover a lot of that in block 4, which will be around the end of July, I think. Okay, so with this block, it's an interesting topic, and I wish there were a whole course in IPAP that talked about immunology rather than a couple just dedicated lectures to it. Because this is really the only time that we, to use the analogy, the military analogy, we kind of blue force things a little bit. We study the friendly perspective. And the rest of this course is all red force. It's all studying the pathogen's perspective and how it exploits and hijacks and controls and deceives the host immune system. So today, we kind of want to get a sense of how things work optimally when our immune system functions properly and does what it's been designed and evolved to do for thousands and millions of years, which is handle all these pathogen threats that we get constantly. On the intro day, when we were first talking about the ubiquity of microbes, every time we take a breath, every time we take a drink of water or eat food, we're constantly being bombarded with different pathogens, right? Unless you're eating like irradiated space food or whatever, or breathing sterile air. It's just natural, right? So our body is used to confronting these pathogens, it's used to dealing with them very quickly and adeptly. But though we'll approach this, are actually going over these ELOs a little bit. We do have some definitional terms and foundational concepts to get through. We'll go through those. And then we'll get more, especially later in the deck, we'll talk about specific characteristics of antigens versus immunogens. We'll talk about the innate immune system first, because that's what your body throws at these pathogens first. It's the first line of defense. So we'll talk about that, the components, these mechanisms, various functions of the innate immune system. And then we will talk about all of these major players. So we'll talk about the cells that do all this work for the innate immune system. And some of these so-called organs, like lymph nodes, organs of the immune system. Then we'll go into the adaptive immune system, which is the more specialized antigen-specific component of the immune system, talk about that, and basically just mirror what we talk about. When we talk about B cells and T cells, we'll spend a lot of time talking about antibodies and how they function, antibodies produced by B cells. We will make sure we understand the difference between the humoral or the antibody response versus the T cell response in the adaptive immune system. Again, talking about the functions, the effective functions of antibodies, which are also called immunoglobulins. And then we'll finally kind of tie all these concepts together when we talk about what it means to be immune, what active immunity is, what passive immunity is, primary immunity, secondary immunity, some of those terms. And then we will talk about dysfunctions and disorders of the immune system, how this can all kind of fall apart as well designed as it is. And this we don't spend a lot of time on, unfortunately, but it's super important. Quick outline, that's basically what I just said. We'll talk about vaccines a little bit, so you have an idea. As we go through this course, we talk about different diseases that we do have vaccines for. At least you have somewhat of an understanding of how those vaccines work, what they do for the immune system, how they prime it. Okay, so the immune system itself is a little different than other body systems that you're going to be studying in, like AP, for example. Like your digestive tract, it's a pretty tidy, discrete system. Your integumentary tract, your nervous system, et cetera, et cetera. When you're talking about the immune system, you were talking about some organ-wide systems, like the skin is an important part of the immune system. Like the lymph nodes are important, the spleen, the thymus, but a lot of this is really at the cellular level. So the immune system, we're talking about those lymphocytes and leukocytes primarily, what they're doing, and their coordinated response. The immune response itself is this entire very complex process by which your body recognizes almost an innumerable number of different threats. Like a mind-blowing number of threats. So it's very hard to teach us, and even if you start reading this, or if you're interested in the topic and you start reading it on your own, the more you read about the immune response, the more you realize you don't know about it. It's one of those very humbling topics that is so complex. So we'll kind of try and keep it simple, but at the end of this lecture about immunology, I hope you also have an appreciation for how complex this topic is. Okay, and I hope this is something you will never be wasting your time on if you continue to self-study immunology. It's always going to pay off in dividends throughout your career. This is how it'll help you manage your patients and help you understand how they're fighting off infections. With immunity itself, that's just referring to whether or not we are resistant to diseases, whether or not we've mounted an immune response and we have memory to how to fight a specific pathogen. This is about as simple of a graphic as I can find that this is a good overview slide of how the innate and the adaptive immune system work together, and some of the major players in terms of the cells. But essentially, this particular figure draws a timeline where you have some sort of microbial insult, and then 12 hours later you see a transition from innate to adaptive immunity. In reality, it is obviously more complex than this, but within a couple hours of, let's say, a bacteria entering the body, let's say you get a splinter that's loaded up with bacteria. Within a few hours, you're going to have a lot of these first responder innate cells recruited into that area, and they're going to be doing their thing, trying to clear those bacteria as quickly as possible from that area. something like splinter, they're probably gonna be able to manage it on their own without any involvement of the adaptive immunity. But let's say that microbial insult were to persist in the area and not be cleared in that time frame. Right around 4 to maybe 96 hours, so up to maybe 4 days later, this is when you start transitioning into the adaptive immune response. So those innate immune cells, those dendritic cells, those macrophages, and eosinophils, and NK cells, natural killer cells, they're going to be secreting cytokines, and those cytokines and other communication molecules are going to be signaling these lymphocytes to be activated and be recruited to that area as well. Within 4 days or so, on until that infection is resolved, it's mostly going to be the adaptive immune response that takes care of the infection from there. It means it's bad enough where the innate immunity, or innate immune cells cannot manage to clear the infection. So at this point, these cells will be heavily involved in clearing the infection, and there will be memory cells that persist, that retain a record of how to fight those particular pathogens. And then the infection resolves, or you die, right? So that's another outcome. And some of these, when we get into blocks 3, 4, and 5, some of these pathogens that we're going to be talking about, they will persist for long periods of time. So it's not like a one and done clearance of some pathogen. It can persist for weeks or months, and sometimes years, sometimes decades. So it just depends on the pathogen. And also, not accounted for in this is the host status as well. So, are you healthy? Is your immune system functioning fully? Are you a baby? Are you a 90-year-old? Are you a 30-year-old? Are you otherwise healthy? All sorts of things on just the host side as well. Okay, so for the innate response, when we talk about innate, what does the word innate mean? Initial, yeah, and intrinsic. So this is what you are born with. It has a couple other nicknames. You don't hear it very often, but constitutive, and then sometimes natural or native immunity. But the key word is non-specific. All of these effector cells are going to be trained to recognize generic patterns, but they don't recognize specific antigens. So as we saw from the last slide, they're also fast-acting, pretty immediate. We have some barrier functions that are important. We have these cells that we'll be talking about individually. And then probably tomorrow we'll start talking about the specific innate processes, not just inflammation and fever, but also like breaking down what does it mean to present antigen, what does it mean specifically to phagocytize something. In terms of the mechanism, going back to that splinter analogy, when you get that splinter in your hand somewhere, for example, how does your immune system know if there's a problem there? How does it see?
Antigen?
Just sees it as a foreign body.
Yeah, some antigens, and specifically, so your immune system, like in terms of sensory input for these cells, there's no sight, there's no sound, but it's all done by receptors. So these cells, some of them are immune cells, some of them are non-immune cells, like just regular epithelial cells, but they have receptors on them that are built in to recognize patterns. We'll talk about those too, but those are toll-like receptors and pattern recognition receptors, PRRs. So like, for example, with this classroom of people, what I'm seeing in front of me are all 25-2 students, right? And I don't know you yet individually, I haven't spent that much time with you, but if somebody walked into the room and sat down in civilian clothes, that would trigger my pattern recognition receptors, and I'd be like, there's a problem here, right? So the pattern there is just the uniform, that's all I'm able to see right now. As I get more exposure to you, I will start recognizing you specifically and individually, and I'll be able to say like, that's a totally different student that was in here the other day, you know? But over time, that takes multiple exposures to advance from that level. We talked about that a little bit, but the type of immune response depends on the pathogen and route of entry. So innate immunity is very, it's responsible for shaping how the adaptive response evolves as well. That's important. And then the connection to the adaptive immunity is conferred largely by triggering and signaling with different kinds of molecules called and some direct actions as well, direct application. The adaptive response, we have a couple other names for that: acquired immunity or antigen-specific immune response. These are characterized by, again, like that word specific keeps popping up, but it means that you recognize only that particular pathogen, and very long-lasting protection, sometimes for the life of the host, you can have protection. These cells are a different cast of characters. Again, we have our lymphocytes, B cells, and T cells. With B cells will produce antibodies. Antibodies have a bunch of different functions, and then the T cells themselves can be stratified into two different groups: helper T cells, which are responsible for activating these cells, and then cytotoxic T cells, which are responsible for killing target cells that they've identified as being a threat. They're on the receiving end of antigen presentation from the innate immune cells, so they're receiving those activating, triggering signals. And then once that threat is neutralized, once they clear the pathogen, and like a small subset of those B cells and T cells will be selected for to persist. Those are the memories T cells and B cells. The analogy there is just like the first time you see primary initial exposure to a threat, you've got to figure out what works to clear that threat, right? So it's like, it takes a long time. It takes weeks and weeks to get to that point. You've got to go through this entire key ring trying to figure out which particular key unlocks that problem. But once you've done that, hopefully you label it, and next time you're confronted with that same problem, you're like, yep, this is the right key for that, to maintain it. So the secondary response, when you get re-exposed to, whatever respiratory virus or bacteria on a splinter, is going to be faster, it's going to be stronger, like more immune cells being produced, and it's even more specific. This table is more or less just for you guys to study, but it sums up what we just talked about when you're comparing and contrasting the innate and adaptive immune responses. Specificity, again, like not very specific for the innate immune response. It's just like a curmudgeon, one force instrument. Adaptive is much more surgical precision of which microbes to target, high level of specificity. The diversity of recognition. Innate effectors, this is on a genetic level, so it's beyond the scope of this course, but they don't have the ability to recombine genetically those different components like T cells and B cells do. So their diversity is basically categorized into those motifs, like recognition of something weird, some weird pattern. Double-stranded RNA, that's weird, gotta respond to it. Chitin, that's weird, that's bad, gotta recognize and respond to it. But again, with adaptive immune cells, exact recognition of those antigens. Memory is present with the adaptive immune system. In either case, non-reactivity to self, both of these arms of the immune system should be non-reactive to self. What does that mean if you're non-reactive?
Is that, it has to go with autoimmunity?
Yeah.
It's under a target cell.
Exactly. So what happens if you have an overreactive immune response? Problems, yeah, autoimmunity, like a trigger-happy immune system is not a good thing. Fratricide, causing problems. The components are a little bit different with these two different systems, but we'll talk about those more. Secreted proteins, innate immune response will secrete complement cytokines. The adaptive immune response with B cells will secrete antibodies, and those cells will also secrete cytokines as well. And then again, just different cells involved. This figure is probably too basic, but it is essentially just showing you what we talked about, where you have this exposure, and if your innate immune system handles it, you don't get any disease at all. Just quietly deals with whatever pathogen you're confronted with. If you do go on to develop a disease, your adaptive immunity gets involved, you recover hopefully, and then you mount that specific immunological memory. So if you're confronted again or re-exposed, you are immune, or you never use symptoms. Right. So, before we get into this slide, I think it's pretty early, 10:35, but why don't we go ahead and take a 10-minute break and finish off up to the rest of the cells today, so we'll just keep it nice and light, okay? So yeah, 10-minute break, 10:45, we'll start back up again.
Is there any way to edit the I don't know how to use what this is, like, I use it or
I think that's how much.
yeah, but it's just
So the state parking. Okay, let's say, let's use a specific example, say respiratory virus. Say coronavirus. This is the first time you'll ever experience a virus where your immune system is starting from scratch with innate and adaptive. When the adaptive immune system goes under, you'll have those memory cells, right? Those memory cells are important to functioning as
okay, so you said
Yeah, see if like this one,
you had to try to what you're going to do is just like
just, oh, you know what I mean?
allows the cells to actually go out and
Yeah, I know they have like size.
that's
there's no
that's a big detail. I didn't think that happened. Technically it did, but
Well, I thought it'd be cool if there was any.
so this is
well, possibly, but if I could think of one, we have specific things on
This kid's like, it's like, no sense, like, yeah, like there's just none. Where like the daughter was like, she was very aware. She knew like what was about. Just hard study about. We can just study math, right? Dude has like probably concussions.
hasn't been
Just the way he does it.
afterwards, I said,
It's like about 5, it's like one thing. And it came out and just hangs by his feet and stuff. And his head is technically right above the constantly. Yeah.
they do actually,
Definitely.
similar,
Yeah.
so it's
My wife.
responsible for actually giving them
Because they don't like it.
Yeah.
also a lot of other,
This is what I did.
CD4 cells are super important for
Based on.
Or is the innate adaptive, there's different factors to this, and there's different factors, different cells that are used to adapt. It's like you let's get back. It's just structures, yeah, it's just like, this is what we're now how big different shapes. I think they both are.
You just really seems to be.
Yeah.
Or just find a page that breaks that. You know what I mean? So just this, basically one first. Yeah. Or I can do all of these things. Well, yeah, you could save it at the end.
It's already fine.
See how it size. One.
You can fail.
I want one.
For right now, it means all of it. That's dirty balls, dude.
Go back to that. Go back to your last folder. No.
Okay, guys, let's pick back up again. So, you know when you're done being done with the immune system, Carl will finish off, we'll talk about complement and some of those processes, but I do want to get through all this stuff today. We'll talk about physical chemical barriers of the innate immune system and get that introduction to those cells and basically like what these primary innate cells do, what their functionality is. So, it's not working. So barriers. First we'll start off talking about those physical barriers of the body. The skin's kind of the obvious one. The skin itself is designed in such a way as basically to be ultra protective against different pathogens. It is such a good barrier that really there are no bacteria that can directly penetrate intact skin. really no viruses I'm aware of either. There are some parasites, like some worms that can burrow into intact skin. But for the most part, it's a very, as long as it stays intact, it's a perfect barrier function in and of itself. When you have any kind of break in the skin, of course, that's how bacteria can get in through wounds. But even then, you have a bunch of immune cells that surveil close to those top layers of the skin that control that area and respond very quickly. For the skin itself, that outer layer, is that alive or dead? Your outermost layer of skin? It's dead, right? Those are stratum corneum, cornified keratinized cells that are just loaded with keratin, right? And they get shed constantly. So the few bacteria that are adapted to living on the harsh environments of your skin, they don't stay there very long. It's pretty transient. It gets sloughed off constantly. It doesn't matter how much you shower, like your body's just shedding those keratinized skin cells all the time. Other qualities, of course, you sweat. And through your sweat, you have a bunch of antimicrobial peptides in your sweat. You have lactic acid, which drives down the pH, keeps it pretty acidic. So again, like those bacteria that are adapted are generally able to tolerate low pH conditions. It's dry, bacteria like their moisture, they like their humidity. So that's one of the reasons why the drier parts of your body don't have a lot of microbial populations, but the moisture you get, the more bacterial things get. And then also you have competition. So just the fact that you're colonized on your skin, it means that it's a selected niche for other bacteria as well. So that's the skin. In terms of other ways into the body, assuming your skin stays intact, what are the main portals of entry, do you think, into the body? Mouth, nose, 100%, absolutely, yep. Yep. Yeah, watch out. Urogenital tract, right? STIs, the word. Did I even mention the eyes? Eyes, right? I mean, a lot of respiratory viruses, bacterial pathogens can enter through the eyes. So all of these different portals of entry require like heavy, up-armored immune presence in order to combat that. These mucosal surfaces, they are specialized and ready and adapted to meet constant stress, especially in your airways and your mouth where you're drinking water, drinking fluids, eating, and breathing in air, like lots of microbial threats. And those microbial threats, of course, oftentimes are carried all the way down to the intestines, which is like its own unique ecosystem for the microbiome. So these mucous membranes themselves, in addition to those immune cells that are heavily populated those areas, all of these different areas, mucous membranes will have secretions. Those secretions will have chemicals, we'll talk about a little bit more in the next slide, antimicrobial chemicals, very non-specific but effective. And then we talked a little bit about cilia the other day, but particularly in your respiratory tract, you have respiratory epithelial cells that are ciliated, that if you're healthy, should be functioning to beat upwards and remove those pathogens. And that's why smokers have such a hard time, right? Because those ciliated respiratory cells are just destroyed in the process over time. An example of mechanical removal in addition to the cilia would be something like urinary flow. If you have bacteria that are trying to attach to the epithelial cells of the urinary tract and establish a urinary tract infection, they better have those adhesions in place, otherwise they're going to get flushed out through a high pressure urinary flow. That's the whole point of that. And there are other examples as well in the body. Body temperature too is super important. How does that impact bacteria and viruses?
High temperatures denature proteins.
Say again?
Like when you get an infection, you get a fever, so it denatures the proteins.
Yeah, exactly. So if things get really bad and you have a systemic response, you can have a fever, and that fever does so many things that are very, very effective at removing microbial threats, but are also miserable for you, right? But In general, the immune cells themselves function better at those higher temperatures, and those pathogens suffer a lot at those higher temperatures. So however bad you have it, those pathogens have it much, much worse. Every degree increase in body temperature makes it that much harder for those pathogens to survive.
Is that the same for viruses too, or is that bacteria specific?
No, that's the same for viruses too. When you have a viral infection that is kind of getting out of control and you need a systemic response, fever will suppress viral replication, and it will also, again, like promote immune cells like cytotoxic T cells, for example, that are designed and adapted for clearing infected cells. So fever is actually really, really important for clearing viral infections as well. Okay, and then chemical barriers, hand in hand, we have secretions from the eyes, secretions from all of, saliva, nasal secretions, vaginal secretions, all of those different secretions in the body have antimicrobial peptides in it, including lysozymes, which are, you've probably heard of present in tears. Those are antimicrobial in nature as well. You have a specialized molecule called defensins that are small positively charged molecules that work approximately by, this is very, very rough, but because most bacteria have an overall net negative charge on their surface, they will insert themselves. They have amphiphilic properties, so they have hydrophobic and hydrophilic regions, and will insert themselves into that bacterial cell wall and basically cause that bacteria to lyse. We also have other hostile conditions throughout the body, throughout the gastrointestinal tract. The pH changes dramatically. When you are ingesting food or water, when it hits the stomach, what do you know about the pH of the stomach? Super low. What is like that pH ballpark? Yeah, it's like maybe 1.5 to 3.5, average like 2 or 3 pH. Super, super acidic. The body does not want to be colonized by bacteria in the stomach. So we don't really select for or tolerate bacteria to colonize our stomach. There's a couple exceptions, like Helicobacter pylori is one that can tolerate super low pH. But that pH is low enough to prohibit colonization from the vast, vast majority of bacterial threats. Bladder and kidney is much less so. The pH is higher there, but still, conditions are pretty inhospitable for bacteria. The intestinal bile, that probably shouldn't be grouped under acidic environments. That's actually a little bit more on the basic side. It's meant to neutralize the pH of stomach contents going down into the intestinal tract. But nonetheless, it's still not a great environment for bacteria to be in bile. See if I missed anything there. So, okay. In terms of the cellular players, we have a bunch to get through. We'll talk through most of these, but this slide is showing you a lot of information, starting from where all these different immune cells we're going to be talking about, where they come from. So they come from stem cells. Stem cells are special because they're undifferentiated cells that are uncommitted to a function that they will someday have. Where in the body do we have our stem cells as adults?
Blood marrow.
Bone marrow, yeah. That bone marrow is super important for producing all blood cells, not just the white cells, but the red cells as well. Hematopoiesis, right, the generation of blood cells. Once these stem cells start differentiating in the initial rounds of differentiation, you have pluripotent stem cells which are capable of further differentiation. Whether or not a pluripotent stem cell becomes a lymphoid progenitor or a myeloid progenitor depends on what kind of signals it receives. But if it receives signals to differentiate into a myeloid progenitor, it will become either an immune cell or it can become a red blood cell, an erythrocyte. Your bone marrow produces a crap ton more red blood cells than it does any of these white blood cells, any of these lymphocytes here. In this microscopy image of a field of blood cells, what are the most of these cells in this field? Yeah, these are all red blood cells. So if you look at the way they're quantified, per microliter of blood, you have between 4 to 6 million million erythrocytes, or RBCs, versus the total number of white cells, or leukocytes, is about 1,000 times less, right? 4.5 to 10,000 white blood cells per microliter. So you have tons and tons more erythrocytes being generated. This is a stain called a Gibbs stain. If you've ever worked in the hematology section before, anybody ever done like a Gibbs stain or what it is? A few people. Okay, so you use a Gibbs stain. It has acidic and basic staining, and it allows you to look at the features of some of these leukocytes, because the way they stain will tell you whether or not they are, for example, a neutrophil, like this guy over here, or a lymphocyte, like this cell on the left. That's a lymphocyte. Staying with that myeloid line for a second, I'll come back to the lymphoid line. With the myeloid cells, they will further differentiate into these CFUs, these are colony-forming units, and then they receive more signals to differentiate. With those white blood cells, we have basophils, eosinophils, we have mast cells, which are not on this slide, but that's another type of granulocyte, or a cell with granules in it. And then from this progenitor, which is called a granulocyte, we have either neutrophils or monocytes. Monocytes are important because they have further differentiation. They can become either dendritic cells or macrophages, which have different functions. On the lymphoid side of the house, B cells and the T cells are the ones that I was talking about that are important for the adaptive immunity. Natural killer cells are also this lymphoid lineage. However, they are part of the innate immune system. So by definition, that means that they have a non-specific way of recognizing antigens and microbial threats. And this is kind of just an FYI, you definitely don't have to memorize it, but it's good to know. This chart up here is from the textbook. It gives you an idea of like what the relative distribution of these different white cells are for microliter blood and what the normal range is. Okay, so we'll start off with macrophages. Transmissions, electron micrograph of what a macrophage looks like. Very big cells relative to the other immune cells even, which tend to be large cells to begin with. They have a pretty high cytoplasm to nucleus ratio, meaning that if you look at this cartoon of it, they have a smaller nucleus. It looks huge, but it's actually on the smaller side compared to some of the others that we'll be looking at. One of the key features, a couple of key features, one, they are extremely long-lived for immune cells. They are meant to be residing in tissues, doing surveillance in tissues, and most of the time they are fairly mild-mannered. They just exist in tissues, sampling, going about their daily business, making sure that there are no pathogens in that tissue area. However, if they do encounter during their surveillance and sampling of whatever is going on in their environment, if they encounter one of those patterns or motifs that's indicative of a pathogen, they take on a whole other character, and they become like enraged, angry macrophage. They kind of hulk up, they actually physically increase in size, and they become aggressively committed to phagocytosis. Phagocytosis is just like it sounds, from the Greek roots phago, which means eating, cyto, eating cells, and the process of osis. So professional eaters, there are other cells that phagocytize targets, but these are the very best. They are specialized for phagocytosis, based on just receiving those like patterns that are indicative of some threat. So they engulf and digest these different pathogens, bacteria and other things, cellular debris as well. Like when your normal cells just die off, macrophages will come along and clean up that mess too. So just keeping everything nice and tidy. But if it's a microbial threat, they have another important function in antigen presentation. So they don't just digest that pathogen, they also tear it apart and present pieces of it on their surface. So those pieces can be shared with the adaptive immune response. They can build that specific immune response. They also have another important role in cytokine production, where you start off in a localized infection tissue, let's say you get injured, you wanna make sure that you can handle it, and it's not too big of a job for you to handle locally. So when you secrete cytokines, that's basically telling other cells in the immune system to come to that area and help out so things don't get out of hand. It's a very, very important function also. And they also do things like tissue remodeling. A lot of different tissues have specialized types of macrophages, specialized glial cells in the brain, you have Kupffer cells in the liver, alveolar macrophages in the lungs to help clear infections. So they get tissue specific, but their overall function is basically like clean things up, make it tidy, and share that information through antigen presentation, cytokine production. Make sure other cells are aware that there's a threat in that area. This is such a wordy slide. Another important cell are neutrophils, also known as polymorphonuclear leukocytes, because of this very weird nuclear architecture they have, multi-mode nucleus. So PMS, in terms of relative distribution in the blood, neutrophils make up the vast majority of your leukocytes, between 40 to 70% of the circulating leukocytes. Because they're circulating in the blood, because there's so many of them in the blood, they're designed to be recruited very quickly. So they receive some of those distress signals and cytokine signals, and very, very quickly, these things will squeeze into any tight space in the body through a process called diapedesis, squeeze through tight spaces, and they're ready to respond to those infections. In terms of their capabilities, you will notice that there's some redundancy with macrophages, and that's common with immune cell function. There's a lot of redundant capabilities. So they too are capable of phagocytosis. However, unlike the macrophages, which are long-lived, remember they're there for weeks or months, these guys are extremely short-lived. So when they phagocytize a bacteria or a virus or whatever, they don't really get involved in antigen presentation. Basically, their job is to destroy that pathogen, and then they usually die afterwards. So, short-lived, you're producing huge numbers every single day, 10 billion cells a day, but max cells circulate for a couple days, and if they do encounter a threat, they die. They commit suicide, program suicide, immediately after that encounter. And that's fine, because they're just regenerating and replenished very quickly. By the way, what is the term for programmed suicide? Apoptosis. Yep. That's another thing we're talking about repeatedly in this course. So they're fine with dying, little kamikazes, very, very dangerous cells. Your body seems to know that they are dangerous and control them tightly for that reason. But when you have immunopathology or problems caused by the immune system, these guys are major contributors to that. They do cause a lot of problems, and that has to do with their mechanisms of dealing with microbial threats. Phagocytosis is a pretty clean way of destroying something. It's not very problematic. But with the second mechanism that they have called degranulation, they use these granules, which are these kind of electron-dense areas you can see on this electron micrograph, these dark patches. They use those granules to poison target cells. So they form a synaptic cleft, and they degranulate and release those granules, and those kill those target cells. That's a sloppy process that causes additional problems. And when these guys die, they die in an uncontrolled manner. Those granzymes get out, and that also causes problems for the host. So this is kind of like, think collateral damage when you think about neutrophils. That third mechanism is probably the most damaging mechanism. It's called, and I love it though, it's super interesting, super cool. It's called netosis, which is a play on apoptosis. But NET stands for neutrophil extracellular traps. And what happens here is these cells go straight to suicide mode. They recognize that there's a pathogen. They start condensing their nuclear material, their genome. They break down their nuclear envelope, and then they kind of just rupture, their plasma membrane gives way, and super kinetic event, but they just hurl their DNA and their proteins at whatever target is in their direction. And of course, they die in the process, but this cartoon is kind of showing you what is happening there. You've got a bacterial cell, and this neutrophil is just seizing its contents all over this target and completely destroying it in the process. Problem with this is, what do you think happens if a neutrophil commits mitosis and it's around some other like host tissue, host cells? Yeah, it'll cause damage. So if this is happening like in a blood vessel on a wide, large scale, those endothelial cells of blood vessel are going to be torn up. And if you get a lot of that, you can have some serious damage, serious damage, internal bleeding, things like bleeding disorders. So, very, very dangerous, but these guys do the job. They are very, very effective in clearing bacteria and other pathogens quickly. Large component of pus, of course, like if you have an infection, you get that pus coming out. It's not just neutrophils, but a large part of that is other dead bacteria, other immune cells, and lots and lots of neutrophils. Yes, sir?
Is there a tier list of when you'll decide to use phagocytosis?
Well, that's a great question. I don't know, but probably. My gut instinct is if whether or not they deal with a different pathogen by phagocytizing it or poisoning it or necrosing it depends on maybe cytokine signaling that might determine it. I'll look into that because I'm curious. It's a great question. I don't know. Chemical. That I said sneeze because in my mind I sort of analogize it to like a bunch of sticky, gross stuff being used to trap those bacteria. And I think it's degradative as well. So if you're like one lonesome bacterial cell that is in the direct line of sight of mitosis, you are covered in this sticky, decondensed, granular material and completely trapped. You'll probably just get degraded afterwards. Yeah.
So it's not like it's sneezing peroxisomes or lysosomes. It's that granule
The stuff that actually gets released is probably most of what is in this cell. So histones, lysosomes, all of that nastiness is going to come out of the cell. And that's stuff that you kind of, again, like you don't want it to be released so much because it's also bad for the host. And somebody had something over here. Nothing. Okay. I think that's everything I'm going to cover on this slide. You can see there on that game sustain very clearly. They're pretty distinctive, easy to pick up if you're looking at what's nearest. Okay, another phagocytic cell are dendritic cells. Much less prevalent than some of the other cells that we talked about, especially with the neutrophils. Less than 1% of all those circulating white blood cells are dendritic cells. They are phagocytic, but not nearly to the extent of macrophages. What they specialize in is this antigen presentation, APC, antigen presentation cell. Form follows function, so if you look here, you look at all these tentacles that are coming out of that cell. That's why they get the name dendritic cell. Dendrite is like a tree branch, right? It's Greek for tree branch. So they send out all these tendrils, and they are basically sharing that information, doing antigen presentation with T cells and other immune cells. So that's what this is showing. Even on this micrograph in the bottom right, I think they experimentally made these dendritic cells take up some like phosphate beads or something like that. That's why it's labeled P. But you can see all around the periphery of the cell, these little tendrils coming out all over the place. Those tendrils are responsible for like squeezing into small spaces and capturing different antigens and then being taken into that dendritic cell. And then it just gets processed like any other antigen. It gets processed inside. We'll talk about more on that tomorrow. Processed and then presented. So the very best antigen presenting cells in the immune system are dendritic cells. That is what they are dedicated to do. But, you know, they also phagocytize as well, and you have to phagocytize in order to do that. They will migrate to the lymph nodes in order to be in that very busy center of activity and share that information maximally. So that occurs mostly in the lymph nodes after they pick up these antigens. And just a very, very important part of activating T cells and starting to get the adaptive immune system on board with a specific antigen threat. Real quick, on neutrophils, did I mention that these are not antigen-presenting cells? I did, okay. They're too short-lived, basically, and they don't express MHC molecules on their surface, which we'll come back to later. Or they don't express any of that, I should say. Okay, for these cells, I threw them all on the same slide just for the sake of simplicity, but these are all granular cells. You can see, again, those dense areas on these micrographs, but these are granule-dense parts of these cells form Gall's functions. What they do is release these granules and kill different types of cells. With eosinophils specifically, they are super important in clearing parasitic infections. So not just protozoans, but an entire gigantic worm in your body can be taken down by enough eosinophils that are releasing these poisonous cytotoxic proteins. The main one is called major basic protein, MBP. It's not a great name, but it's a super important protein for killing those worms. These mature in the bone marrow. Relatively small percentage of leukocytes are eosinophils, but they can be also upregulated when they confront like a parasitic infection. Very distinctive bilobed nucleus, pretty easy to pick these out in blood smears, what they look like. With basophils and mast cells, these are a little bit harder to distinguish, but they still have that very grainy appearance. They're important in inflammation. Both basophils and mast cells are super important pro-inflammatory cells. They have roles in allergic reactions as well. If you suffer from allergies, you've probably heard about these cells, the fact that they release histamine, which causes a lot of the symptoms that you associate with allergic reactions. But in terms of differences between basophils and mast cells, they're pretty similar. They're both really, really rare, especially mast cells. You almost never see them, very, very rarely. You do not find these guys in circulation. They hang out in blood vessels, and that's kind of the main difference between mast cells and basophils. And then later we'll talk a little bit more about its receptors for a specific class of antibody. Okay, and then lymphocytes, these are the cells that are primarily in the adaptive immunity, adaptive immune response, but I figure since we're talking about cell types, we might as well cover them here. This is what a lymphocyte looks like. Again, we cannot tell the difference just looking at this blood smear whether this is a B cell or a T cell. We'd have to probably do something like an immunoassay or flow cytometry or something like that to know if this is B cell or T cell. But functionally, we've already covered this. B cells are the ones that are producing antibodies, and T cells are the ones that are either activating other immune cells or cytotoxic, if they're CD8 cells. And then we have natural killer cells, which you also can't really morphologically distinguish from these other lymphocytes we'll talk about on this slide. You have 2 to 5% of your leukocyte population made up of these natural killer cells. A slightly higher percentage for lymphocytes, but they're not quite as abundant as T cells. In terms of what they do, what do you think they do, given that name? They kill. Yeah, natural killer cells. And they kill in a way that is very similar to how cytotoxic T cells kill, which isn't surprising given that they're both of this lymphoid lineage, right? However, there are some differences. If I said that natural killer cells were innate cells and cytotoxic T cells were adaptive cells, what do you think should be the main difference in their killing mechanism? Yeah, exactly. They're recognition of the problem in the first place. So they have a different way to recognize threats, and it's kind of beautifully complementary how the blind spot of T cells covered by a natural killer cell. This cell has a tendency to like kill first, ask questions later, err on the side of killing something, as a good innate cell would do, right? So it's a very cool cell. We'll talk a little bit more about which kind of receptors and signals it receives to kill, but suffice it to say for now that it's permissive, and it happily kills without any kind of prior sensitization, meaning it doesn't have the same signaling requirements needed. In terms of its mechanism, it releases cytotoxic granules, the same way that cytotoxic T cells do, and it can also induce apoptosis as well. There's a really, really cool electron micrograph of a natural killer cell with a TC being target cell, not T cell, but target cell. So this is maybe a cell that's been infected with a virus or some other intracellular pathogen. And this natural killer cell has like half dozen or a dozen different interactions that are going on in between it and that cell, where it's receiving all the right signals that I'm going to kill this thing. And indeed it does. It's so cool. Okay, I know it's super early, 11:20, but I think we'll call it a day there, and we'll pick up tomorrow talking about complement and how cytokines work, and a little bit more about antimicrobial peptides. Any questions?
I know you said you're not familiar with pop culture. The burning question in my head, have you seen Osmosis Jones?
No, I have no idea what that is.
So, Osmosis Jones is a half cartoon, half real movie, where it follows a white blood cell, and it's trying to fight. It teamed up with like this generic pill, and they're trying to fight a virus.
Cool.
Inside the body.
Oh, I love that.
I watched it as pre-study for this class.
Okay. Is that like how most of you guys did your prep work? Okay, I'll put that on my list of
Osmosis
like,
Jones.
yeah.
Now, be prepared. It's like 90s, maybe early 2000s quality. So.
Okay.
Bill Murray.
Oh, awesome.
Yeah, he eats an egg that like a monkey went to his mouth
Yeah.
and then dropped on the ground and then he eats it anyways. Despite his daughter's best efforts. And
Okay.
he gets infected. I think Ebola? Anthrax?
Oh, weird. So it's like, okay, so he eats it, so there's probably like, you would be thinking it's some like enteric pathogen, but anthrax?
He eats a raw egg, yeah.
Weird. Okay, I have so many questions.
And I want you to
Yeah.
kind of like,
Oh, I'll give it the full, yeah, the full review. How, is it a series or like one?
no, it's one.
I don't know if I can handle multiple and actually.
I'm not sure. I'm pretty sure that he's like watching the series. Chris Rock is the voice of White Blood Cell.
Oh, cool. Okay.
It's like a comedy, but
Okay.
yeah.
Yeah, I'm curious like which immune cells are represented in this little weird like, foodborne anthrax scenario. Egg-borne anthrax. It's intriguing. All right, Osmosis Jones for me. For you guys, just look over the slides and or watch Osmosis Jones if you haven't seen it. And I'll see you guys tomorrow at 10 o'clock. Have a great rest of your day.